Welcome to Easy Microbiology. Good morning everyone. Today I will be discussing Corynebacterium ulcerans and how it interacts with its environment and its hosts. Let's get started.
Corynebacterium ulcerans is a rod-shaped gram-positive bacterium belonging to the Corynebacterium genus and having close genetic ties to Corynebacterium diphtheriae, the causative agent of diphtheria. Despite being a rare bacterium, its potential pathogenicity is under investigation, as it has been identified as a probable cause for human illnesses, such as skin infections. C. ulcerans is a frequent bacterial strain that inhabits the respiratory systems of animals such as pigs and cattle. It is also found in soil and water, giving it a wide-ranging habitation. Comprehending its presence and its dissemination methods is essential to preserving human beings, animals, and the environment. This is the morphology of C. ulcerans, a rod-shaped bacterium. It has a club-shaped appearance that is a result of an enlarged and rounded end. It's approximately 1 to 4 micrometers in length and 0.5 to 1 micrometer in width. This image gives us a better look at the size and shape of this bacterium.
C. ulcerans grows well on blood agar and other culture media, forming small, grayish-white colonies. It is an aerobic bacterium and grows best at temperatures between 25 to 37 degrees Celsius.
C. ulcerans is a facultative anaerobic bacteria, meaning it can survive in both oxygen-rich and oxygen-low environments. It is capable of fermenting glucose, maltose, and lactose, and is able to produce the enzymes catalase, urease, and nitrate reductase. Humans are not the original hosts of Corynebacterium ulcerans. The bacterium's primary reservoirs are domestic animals such as pigs and cattle. However, C. ulcerans has been linked to infections in humans, particularly those with weakened immune systems. While C. ulcerans is not considered as a widespread pathogen in humans, its emergence needs to be monitored and preventive measures taken to reduce the risk of infections. The bacteria C. ulcerans has several virulence factors that enable it to cause infection. One of its most significant factors is the production of a toxin that is similar to the one produced by C. diphtheriae, the microorganism responsible for diphtheria. In addition, C. ulcerans produces other compounds that allow it to adhere to and invade host cells, giving it the ability to cause infection. C. ulcerans is a particular offender of the growing problem of antibiotic resistance. It can develop resistance to antibiotics by producing enzymes to degrade them, or by utilizing efflux pumps to expel antibiotics from the cells. This renders effective antibiotic treatment more difficult. Epidemiology plays an important role in understanding C. ulcerans infections. Infections of this kind are not common in humans. They occur mainly in those who have close contact with infected animals, or their habitat, like farms and wet environments. We should continue to be aware of the risk, and take preventive steps to ensure the safety of ourselves and others.
C. ulcerans can cause a range of infections in humans, including skin infections, respiratory tract infections, and invasive infections. The severity of the disease can range from mild to severe, depending on the host's immune system and the virulence of the infecting strain.
We have much to learn about the immune system's response to C. ulcerans infection. However, what is known is that it produces certain virulence factors which allow it to adhere to and invade host cells and thus evade the immune system. Gaining a better understanding of this process can be done by examining bacterial growth in a petri dish. With the correct tools, further immunological responses can be identified and used to develop effective countermeasures.
C. ulcerans infections can be diagnosed through the isolation and identification of the bacterium from clinical samples, such as blood, sputum, or wound swabs. Laboratory tests, such as polymerase chain reaction, PCR, can also be used to detect the presence of the bacterium.
To prevent C. ulcerans infections, it is essential to steer clear of infected animals and their surroundings. Furthermore, practicing good hygiene such as regularly washing hands and food preparation is highly recommended to decrease the likelihood of spreading this bacterium. Antibiotic treatment is the most effective way of treating ulcerans infections. Commonly prescribed antibiotics include macrolides, tetracyclines, and penicillins, which are generally taken for several weeks. In more serious cases, hospitalization may be necessary. By providing appropriate treatment, it is possible to reduce the risk of serious complications or even death. C. Ulcerans is a rare but potentially serious bacterial pathogen that can cause serious infections in humans. To prevent infection, good hygiene practices and avoidance of close contact with infected animals or their environments are necessary. Therefore, we should be careful when dealing with animals in their habitats to avoid infection. Thanks for your attention.